So, students, we are going to study estimation of serum cholesterol. This is our aim for the practical course. Let's know some general information about the cholesterol test. Cholesterol is a organic molecule. So, organic molecule means they are made up of carbon, hydrogen, and oxygen, and it is a sterol, which is a type of lipid. Sterol is a ring, steroid ring. There are steroid ring in the cholesterol structure. In biochemistry, when we study biomolecules, you might have studied biomolecules in class 12 and even in BSc MLT or Diploma MLT. So the structure of cholesterol, there are carbon, hydrogen, oxygen atoms, and it is a kind of lipid. Okay, it's a derived lipid. Cholesterol is biosynthesized by all animal cells. This is what you have to understand, that this cholesterol is synthesized in our body. Our body has an enzymatic machinery, and through biochemical reaction, we can synthesize cholesterol from carbohydrate and from the lipid and from protein also. Okay, it is an essential structural component of animal cell membrane. If you study the molecular structure of animal cell membrane, so you'll be able to find cholesterol is one of the essential structural component. Cholesterol provides, structural integrity to the cell membrane. Okay, and without cholesterol, we cannot make proper structure of the cell membrane. Okay, so this is very essential component. Cholesterol is also one of the precursor for steroid hormone synthesis. Steroid hormones are like testosterone, estrogen, progesterone. And in addition to this steroid hormones, vitamin D, you might have heard about vitamin D, which is synthesized in the presence of sunlight, right? And this vitamin D is very, very important for strengthening our bones. And vitamin D is also synthesized with the help of the cholesterol. So it's a precursors. Precursor means the starting material. Okay, so it's a raw material. Cholesterol is a raw material to make steroid hormones, to synthesize vitamin D in the cell. But excess cholesterol in blood is harmful. Excess cholesterol occur when we take cholesterol from the outside as a diet. Though you already understand that cholesterol is synthesized in our body, so whatever the amount of cholesterol is required, the body can make it. But if you take extra cholesterol from the outside as a dietary supplement, so there can be the excess cholesterol in the blood, and which is very, very harmful, right? We have learned some general information about the cholesterol. Let's know the requirement to perform cholesterol test. If the doctor has written prescribed to go for serum cholesterol test, so we have to collect the sample from the patient, right? And in the laboratory, we require some of the requirement so that we can do the test, manual test. Equipment and apparatus required, like colorimeter, centrifuge, autopipette, test tubes. Colorimeter, with the help of colorimeter, we can take OD, optical density, or we can take the absorbance of the color. The role of centrifuge is it helps in the separation of serum from the blood. Autopipettes, we can use micropipette, autopipette, to measure the specific volume of the sample, serum, or the reagent. Test tubes, where we can add sample, reagent, standard reagent, or blank reagent. Okay. Chemical required for that test is we require cholesterol kit, which is ready-made, and we require distilled water as well for plant. What sample do we need? We need always use unhemolytic serum or plasma. So we can use serum, and it should be not hemolyzed serum, unhemolyzed serum. You can also do the test with the help of plasma, but it should be heparinized plasma. And for collecting, before collecting the sample, fasting for 9 to 12 hours before the cholesterol test is required. So the fasting samples will be preferred. We should ask the patient to report to the lab in the early morning after overnight fasting. So fasting samples should be preferred. Okay. Now let's know the principle of the test. Here, this test is based on the enzymatic method, where we use three different enzymes. We can see CE means cholesterol esterase, CHOD is a cholesterol oxidase, and POD is a peroxidase. There are three enzymes: cholesterol esterase, cholesterol esterase, and peroxidase. Right? So, with the help of these three enzymes, which are present in the reagent, working reagent, we can react cholesterol, and we can make a color end product, and we can measure the intensity of the color end product to know what is the amount of cholesterol in the given sample. First of all, the serum, we have to understand that cholesterol is, some of the cholesterols are found free form, and some of the cholesterols are found in ester form. So there can be cholesterol ester as well. So cholesterol ester means the cholesterol has bound with the fatty acid. Okay. So this cholesterol esters is the cholesterol with the ester of fatty acid. Okay. Both two molecules, fatty acid and cholesterol, has bound together. So in that form, we call them as cholesterol esters. These cholesterol esters are hydrolyzed with the help of cholesterol esterase, and we get cholesterol and free fatty acid. Right. So all cholesterol which are combined with the fatty acids are now separated from the free fatty acid. Okay, we get a free fatty acid from this cholesterol esters with the help of this cholesterol esterase enzyme. And after the enzymatic reaction, the cholesterol which is formed here is oxidized in the presence of CHOD, which means cholesterol oxidase. So the product that we get from this enzymatic reaction is a cholest 4-in-3-1 plus H2O2. So this is the two compound. One is a cholest 4-3-1, cholest 4-in-3-1, and another is a H2O2. H2O2 means hydrogen peroxide. Kim wants to join. Okay. The second reaction, what we have studied, that in the presence of cholesterol oxidase, okay, cholesterol is oxidized to cholest 4-in-3-1 and hydrogen peroxide. Here, this hydrogen peroxide will be used in third reaction. So this hydrogen peroxide here from the second reaction is used in third reactions. So hydrogen peroxide in the presence of 4-amino antipyrine and phenol, okay, and the enzyme is POD, which is peroxidase. So it converts this H2O2 and 4-amino antipyrine with phenol to quinone dye, and this is a colored end product. Okay, so when we get quinone dye, so it produces a color end product, and we can measure the end product through the colorimeter. So more color means more cholesterol in the sample. Less color means less cholesterol in the sample. So color is directly proportional to the concentration of cholesterol in the sample, right? And More color, more absorbance. More absorbance means more concentration of cholesterol. That is what you have to understand. I hope you have understand the principle of this cholesterol test. This test is totally dependent on enzymes, enzymatic reactions, and this is one of the very specific tests. Why we consider this test as a specific test? Because here the enzymatic reactions takes place. The enzyme only recognize its own substrate. So the enzyme cholesterol esterase only bind cholesterol esters. Cholesterol oxidase only bind cholesterol. POD only binds hydrogen peroxide. So this is what the enzyme and their respective substrate. So other component present in the serum will not participate in the this enzymatic reaction because serum contains mixture of the biomolecules. It contains triglycerides, fatty acid, cholesterol, glucose, protein, amino acid, vitamins, minerals. So many organic and inorganic compounds are present in the serum. So those organic compounds does not participate, do not participate in this enzymatic reaction. Okay, so we can say this method is very specific. One enzyme, one substrate. What is the concept we know? If you study the enzymology, then this concept is one enzyme, one reaction. So here the enzymes are uh, enzymes can bind to their respective substrate. Okay. So what we have understand from this principle that cholesterol esters are cleaved by the action of cholesterol esterase to yield free cholesterol and free fatty acid. This is the first reaction we have to understand. That cholesterol esters are cleaved means broken down with the help of cholesterol esterase. And the product is, what are the product? Free cholesterol and free fatty acid. The second reaction is the cholesterol oxidase then catalyzes the oxidation of cholesterol to cholesterol 3,1 and hydrogen peroxide. So these are the two products we got in the secondary enzymatic reaction, cholesterol 3,1 and hydrogen peroxide. Now, third reaction that we have understand that the condensation of hydrogen peroxide with phenol and 4-amino phenazone or 4-amino antipyrine, right? In the presence of peroxidase enzyme, it produces red color quinoline dye, which can be measured at 530 nanometer. So the color of quinine amine dye is pink or red color, okay, and this is the end product, which is a complex produced as a quinoline, and the color intensity of the quinoline is measured at 530 nanometer, or we can select green filter in the colorimeter. So now procedure for the test is we can take three test tube, okay, three cleaned and dry test tube we can take. We can label the test tube as a T, S and B respectively. The T for test, S for standard, B for blank. Okay. And then we have to prepare the respective ingredients in the respective test tube. So these are the ingredients which we have to pipette in the respective test tubes. So working reagent here, we need to add 1 ml in test, standard and blank. In this serum sample, from the serum sample, we have to take 0.02 ml in the test. Cholesterol standard, which is having 200 mg per dl concentration. This cholesterol standard we have to add in the standard tube, the similar volume as like serum, 0.02 ml. And distilled water we have to add in the blank, 0.02 ml. So after preparing these ingredients in these respective test tubes, we have to mix properly, and then we need to incubate at 37 degrees Celsius for 10 minutes. As you know, the working reagent contain enzymes. So in the enzymes usually perform it, higher reaction, higher velocity of the enzyme takes place at optimum temperature, optimum pH. So temperature 37 degrees Celsius, here the enzyme perform at the highest level, and we give 10 minutes, that within 10 minutes the substance end products are produced. Okay, so we incubate at 37 degrees Celsius for 10 minutes, and after 10 minutes we get an end product, which is a quinoline dye, which has a color, and we can measure the color absorbance, okay, at 505 nanometer or 530 nanometer. Now we can calculate what is the level of serum cholesterol. So the formula we have to take, we have to use the formula ODF test divided by ODF standard multiplied by 200. 200 is the concentration of the standard concentration. Then we can write the result in mg. It's a mg per dl. It's not a gram per dl. It's a mg per dl. And mg per dl. That's the mistake. Okay. So result we can use in mg per dl, and the normal range for the test is it's minimum 150 and maximum 250 mg per dl. So 150 to 250 mg per dl is the normal range. You can see that the cholesterol level less than 200 mg per dl is a desirable. If the value becomes 200 to 239, it is a borderline high level. And if the value is more than 240 mg per dl and above, then this is considered as a high level, and it is it is one of the risk for heart disease. Okay. So what is the significance of high cholesterol? If the cholesterol level is less more than 140 or more than upper limit of the normal range, so what is the significance? What happens? So if you have a high cholesterol, you may not experience any symptoms at all. There is no equally any symptoms can be faced by the experienced by the patient. But the high cholesterol levels may result in hardening of the arteries, which is a atherosclerosis. Atherosclerosis means it's the hardening of arteries. The arteries become hardened because of deposition of cholesterol and triglyceride in the wall of the arteries, and because of that, the arteries become hard, and the process by which the arteries become hardened, and that process is known as atherosclerosis. Okay? So high cholesterol level may result in hardening of the arteries, which increases the risk of stroke, heart attack, and heart disease. This is what we can see the cholesterol. This is the normal artery where we can see the white lumen and blood can pass easily. And here you can see the artery which has narrowed. And in the narrowed artery, which is abnormal artery, where you can see the plaque formation where the cholesterol has deposited. Okay. And because of that, the blood supply cannot take place properly. So there is a zigzag mechanism of blood flow. The lumen of the artery has become very narrow, so it will causes turbulence in the blood flow. And sometimes if there is a blockage in this artery, because the lumen has become very narrow, so it may causes loss of blood supply to the tissues, which result in the form of stroke or heart attack, right? So you have to understand that cholesterol test is very important to know the risk of heart disease. Okay, so we have come to the end of the slide. Here we'll know that error analysis, that the test while performing the test, we should know that ecteric and glycemic sera may interfere with the test. Ecteric means if the serum has a high level of bilirubin, so the serum from jaundice patient may so interfere with the test because it has a color. Lipemic sera means if you have taken the serum with the lipid, already the lipemic sera is something like where we can see the whitish layer of uh, serum. Okay, the serum become whitish color. So we need to avoid that. We need to centrifuge to remove the lipemic sera. We need to take the soluble form of serum, the liquid form of serum, the not the what we say. Um, there may be the milky like appearance, and that milky like appearance is a lipid, and that lipid should not be taken for the processing. We need to separate it out, and we can measure the accurate cholesterol in the sample. Ensure complete clot formation has taken place prior to centrifugation. So because we do not take any anticoagulant, So ensure that the blood should should be clotted properly, and after that we can do the centrifugation, and the serum will be separated, and we can process the serum now. Okay. So we have finally come to the end of this presentation. Now we can discuss if you have any questions.
सर इसका प्रिंसिपल एक बार हिंदी में